welcome back. Now, if during all of this you were saying to yourself, hey, T cells, I get it. What about B cells? I thought there were B cells in the immune system too. You'd be absolutely right. So let's go through how they signal. And fortunately, it's a very short little thing. Yeah, so here we go. Here we go. And this is just the B cell activation pathway. There are many other pathways. Many other pathways. All right. What do we got here? We've got B cell receptors. I recognize them. It looks like an antibody and it has a little transmembrane tail. We got those two things that are with the B cell receptor, the alpha and beta chain. We know about those, yeah. Or was it beta and gamma? I forget. No, Ig alpha and Ig beta. Yeah, alpha and beta chain. Fine. I see those yellow boxes at the bottom, so I know those are ITAMs. These are proteins that have ITAMs on them. Sweet. Yeah. And I know that ITAMs can be phosphorylated. So, the idea is that if you get more than one B cell receptor, so B cell receptors are going to need a friend. If they're both binding onto an antigen or an antibody-antigen complex of some sort, or the antigens are all lumped together, because they're close together, this will now bring together several kinases. So BLK, FIN, and LIN are three of the kinases. So I don't know, black or block, yeah. FIN and LIN. These are kinases that are okay at phosphorylating their own ITAMs, but they're much better at phosphorylating the other one. And the more of these you get together, the more phosphorylation you're going to get of those. So by joining multiple B cell receptors together on an antigen, you can get a whole lot of phosphorylation. And then in a B cell, it's not going to be ZAP70 that's doing the job. It's going to be psych. But do you know what psych has? It has two SH2 domains that bind to that ITAM. You know what else about psych? Psych has a kinase domain. Ah, didn't know that. And you know what else about psych? Psych is inhibited except when it's bound. It becomes active as soon as it binds to the phosphorylated ITAM. So there you go. And there's a whole pathway downstream of that. So let us look. So those, and let's just remember it this way. BLK, FIN, and LIN, these are all what we call SRC family kinases, SRC family kinases. So we'll just say a SRC family kinase. We won't have to remember the names because that would be too many names. That would be too many random names. So a SRC family kinase will come along and phosphorylate the B cell receptor. And if we're making additional contacts, so there's a molecule that's called CD21 that hangs out with CD19. Yeah, because whatever, they're about the same age, they can hang out. That will then bind to C3. Remember C3, part of the complement cascade? Ages ago, like a lifetime ago. So if you've got like a bacterium and it's got an antigen that your B cell can recognize, and the B cell is also able to recognize that there's some complement on that thing, then that means that it's got an antigen you recognize and it's got complement on it. This is a bad thing that needs to be, cleared out. And so you'll get the SRC family kinase will start to phosphorylate things. It'll phosphorylate CD19 and it'll phosphorylate the B cell receptor. Here again, CD21 is just a detector. The thing that makes it signaling is that it has CD19 next to it. And it has to have that next to it, otherwise it won't signal. Yeah. CD81 or TAPA1 is also part of that same complex. Yeah. But it doesn't really do anything at this stage, so we don't need to learn it. Yeah. All right, because it's going to be, very like there's the book of Numbers in the Bible, just who begat whom. Yeah, just think of it, kinases all the way down. Kinase after kinase after kinase. Anyway. The SRC family kinase phosphorylates here. That makes a nice little region, which is an SH2 region, which binds phosphorylated tyrosines. That will recruit the PI3 kinase. And what does PI3 kinase do? Why, it takes PIP2 and it turns it into PIP3. So you got this pip2 floating around, and you can pop it off into two pieces, and they can both do all those cool things. Or you can take pip2 and you can turn it into pip3. You can level it up. And once it's pip3, it's now untouchable. And it gets to become the platform for all these other things. So, you remember, right? Remember back when PDK bound to a pip3 and came along and just sort of pushed the little button on AKT and activated it? Yeah, phosphorylated AKT, which then goes on and phosphorylates other things. Do you remember BTK? Actually, no, we didn't learn this part, but it is going to bind to a... this is a different way to phosphorylate PLC gamma. Yeah. And activate PLC gamma. And I think we learned of a different way that does that as well up before. This is what would happen inside of a B cell. The other thing is what happens inside of a T cell. So we're going to assume that our PLC gamma activating, we're going to pretend that's all in a T cell. So learn the T cell version of that. Yeah, life is too short. Of course, PDK is also going to bind VAV. VAV is going to bind to NIC or NOC. NOC or NIC always comes along with WASP. Those two are going to then open up when they run into a CDC42 that's all loaded up with GTP. And once that happens, we will then start polymerizing actin. We're going to bring in those other complexes and polymerize actin. That was like a couple of slides ago. It's there. Put it on some note cards. Get it into your brain. Get this stuff in your brain. Yeah. And so, this is kind of the idea. This is the big summary, and maybe they should have put the summary at the beginning. Maybe I should have been smart enough to undo the way they did it and put the summary at the beginning, but they put it at the end. So, you got your T cell receptor, you got your B cell receptor. They've got proteins attached to them, in this case Ig alpha and beta with the B cell receptor. They haven't drawn the TREMS membrane part of this, but it's membrane bound IgM, so it is there. Here's our T cell receptor with our CD3, all those nice chains, little zeta chains down there. And so, yeah, we're going to get some tyrosine kinases activated, usually by bringing more than one thing together in a zone. And then once that happens, those are going to phosphorylate some adapters and scaffold proteins that'll assemble into little scaffolds that then hold on to other kinases and activate those. And we're going to set off phospholipases and lipid kinases. That's like that PI3 kinase. Those will then set off further signaling, which is going to be our GTPases like CDC42 and RAF and RAS. And also some of these other kinases and phosphatases. They'll then go on to do wonderful things, remodel the cytoskeleton, change the shape of the cell. And yeah, from there, eventually all paths probably wander off and run into kind of a dead end. Well, we know it does this, and we know that at some point that happens, and we're trying to fill in the middle. We're in cell biology territory now, or you could say molecular immunology territory. This is what a molecular immunologist would be studying. You pick one of these pathways and you try and fill in a couple more of the parts. It's a lot of work. It's expensive to do, but it's important. This is the only way we can make wonderful textbooks like Janeway's Immunobiology, version 9. Yeah. on sale now in the bookstore or something, and on Amazon. All right. That's enough for today. See you later. Bye.